I'm Dr. David Seftel, Director of Research and Development for the Macular Degeneration Foundation. And we're here today with Dr. Jaclyn Kovach. And we're going to ask her about her activities and her research interests. Welcome, and thank you for joining us.
Thank you so much.
Firstly, tell us a little bit about your position.
I'm an assistant professor of clinical ophthalmology at the Bascom Palmer Eye Institute, and I see patients in Naples and Plantation, Florida.
And what are your current research interests?
Currently, I'm interested in the genetic basis for age-related macular degeneration as it relates to treatment response for dry and wet macular degeneration and also ocular imaging.
And what did you present at the AAO here today?
At the AAO here today, I'm presenting work that I've done as part of a collaborative effort with our genetics department at the University of Miami and our Vanderbilt Eye Institute counterparts. And we are basically investigating the role that genetics plays in response to anti-VEGF therapy for wet age-related macular degeneration.
What were the conclusions of your current study?
Well, at this point, we have rather a small sample size, but we concluded that individuals that maintained vision had a higher genetic risk profile than those that responded better to anti-VEGF therapy, which is what you would expect.
What specific genes were you looking at?
Well, we calculated risk scores for our patients, two different types of risk scores. We called one the 16RS, which considered 16 genetic factors deemed as reaching genome-wide significance by the AMD Gene Consortium, and also a 4RS, a 4 risk score that considered 4 genetic alleles in complement factor H, C3, ARMS2, and C2CFB.
Now, switching gears to a more broader sense, what is the progress in the area of gene therapy? Are you working in that area?
Well, that's our ultimate goal, but first we need to identify which genes play a role in the etiology of age-related macular degeneration and in treatment response. Earlier, our group collaborated in the AMD Gene Consortium and identified a total of 19 genetic loci that modulate AMD risk. So we're getting close.
There's been some discussion about the use of different types of viral vectors for transporting the genes. What is your current thinking as to the best viral vector?
Well, I think it's as yet to be determined. There are several studies investigating various viral vectors, and we'll have to see which is ultimately best. But our goal in this regard is personalized medicine, personalized treatment for wet age-related macular degeneration.
That's a very important paradigm that we'll be looking at very closely at the conference, and that is the issue of customized care, which was also highlighted by the AAO's work with the IRIS database. Now, you obviously also work with Dr. Jeffrey Goldberg. And what is the level of your collaboration that you're currently involved in?
We're at the same institution. Dr. Goldberg has made many advancements in terms of neuroprotection and in the glaucoma field. So unfortunately, I haven't had the opportunity to collaborate with him much as of yet.
In the area, we've spoken about gene therapy, and then the other area obviously is stem cell. And here there's work going on both in embryonic and adult stem cell, ES or iPS cells. Do you have a level of interest in this area and involvement?
It is very interesting, but I haven't been involved in stem cell research yet.
Some researchers believe that ultimately a combination of gene therapy and stem cell therapy will be a viable approach. What do you think about this?
I agree. Gene therapy would be fantastic because it would offer us the ability to prevent or curtail the disease, and then stem cell therapy to treat the disease hopefully very effectively.
For patients looking at the current progress, where would you say we are in terms of translation? In other words, how long before patients will be able to experience the benefits of your work and your collaborators in the clinic?
Well, I'm hopeful that within the next 10 to 15 years, potentially, we'll be able to translate the work that I'm doing and the work of others into clinical practice.
Thank you very much, Dr. Kovach, for a wonderful presentation.
Thank you.